Welcome back. We're going to continue on here with the digestive system, moving into the liver here, and then continue on through the rest of the digestive tract itself. So the liver is a massive organ that we have in the body itself. It does many things for us as far as its overall function goes, and it's going to provide for um, the body's main way of detoxifying substances that it finds within the bloodstream. So, where do we find it? It is on the right hand side, the upper part of the peritoneal cavity, um, sitting there. And because it is so large, it has several ligaments that help to hold it in place. One being the falciform ligament. This runs along two of its lobes, the right lobe and the left lobe, and helps to connect it superiorly to the liver, or the liver up to the diaphragm rather, and then onto the abdominal wall, so that as you roll around, do somersaults, cartwheels, things like that, the liver isn't just floating all around the abdomen there. We also have another ligament called the ligamentum teres. This one extends from essentially the underside of the liver all the way up to the umbilicus, and this used to be the umbilical vein. So at one point it was a functional blood vessel that went all the way up to the umbilicus and continued into the umbilical cord. Now, as an adult, it remains as essentially just a collapsed vessel and becomes that falciform ligament that's there. It also has connecting into it some of our other connective tissue structures of the abdominal cavity. So we have the lesser omentum is going to take over towards the stomach. That is there. Moving into the underside of the liver, we have the portal system. And so our blood vessels coming from the digestive tract, making their way over to the liver itself. And also situated on the right lobe, we have the gallbladder on the underside of the liver as well, and that is intimately attached into the liver. When we look at the liver itself, one of the functions that it has is the production of bile, and bile is going to be produced in the liver and transported out towards the bile ducts themselves. Those bile ducts are going to make it into right and left hepatic ducts, and then eventually into the common hepatic duct, which then continues on, and its continuation becomes the common bile duct, but it also has an offshoot called the cystic duct. So essentially, kind of as it's coming in from the liver, we have our two hepatic ducts there. We then have our common hepatic duct. It gives off the cystic duct, which then goes out to the gallbladder. And then we have our common bile duct extending downward. And then that's what's going to join then like that blue, the pancreatic duct as well. And then we have the combination of the two as the hepatopancreatic ampulla. That's there. So that's our ductal system for the liver to get bile down into the digestive tract. It all begins in lobules that are inside the liver itself. So these are hexagons in terms of their overall shape. And as the hexagon occurs, it kind of looks like a wagon wheel. And so it has 6 corners to it, something like that. It has a central vein in the very center that is there. And then out in the corners, there are the portal triads. And these portal triads contain vessels, and then they also contain a bile duct. So we've got 3 different structures that are inside each of those portal triads themselves. So we have a hepatic artery, our portal vein, and then we have a bile duct, which makes up the 3 components that are there at the end. Essentially, blood is going to move towards the central vein, and bile is going to move out towards the corners. So it's going to head out towards the portal triads themselves. Running along from the portal triads to that central vein, we have our sinusoids. And so this is where all of the blood coming into the liver is going to essentially become inspected as it moves along. So this is where we can do our detoxification. This is where we can do our dumping of things into the bloodstream as far as liver products, proteins, things like that. And then we can also clean things up via things like Kupffer cells. So these are resident macrophages that are always inside the liver and inside those sinusoids themselves that are there. The hepatocytes have a dual function in a sense in terms of their overall work that is there. One is to deal with the things that are inside the blood, and the other is bile production. So processing all of our bloodborne nutrients, so everything that comes in from the digestive tract gets processed there. It's going to take in things like fat-soluble substances and store them in the liver. And then it's going to take anything that has made its way into the bloodstream, whether it's from the digestive tract or respiratory, and it's going to do detoxification of those substances as well as it comes through there. So all of that is occurring in those hepatocytes, which line the sinusoids themselves. And here we can see a close-up of a single lobule that we have. So we have our central vein at the center of our lobule. At the edges or the corners, we have our 3 vessels. So we have a bile duct, we have a vein, and we have an artery as it's coming in from there. So the arterial side is coming from general circulation. The venule side, in terms of that portal vein, is coming from the digestive tract. So we're mixing essentially oxygenated and deoxygenated blood, and that blood is then going to travel down along the sinusoids and make their way into the central vein. So remember, back to blood vessels, the sinusoids are a type of capillary, which are very, very leaky, allows us to do all the things we need to in terms of the blood itself. As we then manufacture bile, that bile then is heading out towards the bile duct and out. So the hepatocytes are essentially taking all of the bile salts from the blood itself and dumping it into the bile ducts there. So what do we have in bile itself? And so one of the major things that we have there that gives it its kind of yellowish green color that we have in there is the remnants of red blood cell breakdown. And so we have bilirubin. And so part of that is there is going to be excreted, and we're going to lose that into the digestive tract. That's the whole point of the loss of that bilirubin, is to get it out of the body. So bilirubin came from the breakdown of hemoglobin. We had globin, we had iron, and we had the heme component. The heme component is converted into bilirubin, and that becomes a substance that we must get rid of out of the body. And so we have to eliminate bilirubin from the bloodstream. Otherwise, this is going to eventually lead to jaundice within the body itself. One of the other big factors when it comes to bile is that it allows us to emulsify fats. So this is going to allow us to absorb fats in from the diet. As we get them coming in, we require bile in order to help with that absorption process. So here we have a diagram of the biliary tree. And so as we can see, again, that production comes down from the liver, from the lobes, comes into our common hepatic duct. We have our cystic duct. We have our common bile duct making its way down into the small intestines there. So as we detect fatty acids in the digestive tract in terms of the stomach and then the small intestines, that's going to stimulate the release of cholecystokinin and secretin. The cholecystokinin is going to feed back into the gallbladder, causes the gallbladder to contract. As the gallbladder contracts, that pushes and forces the bile from the gallbladder down through the cystic duct into the common bile duct and out into the duodenum. That's there. The secretin portion travels up to the liver itself and essentially makes it so that we have an enhancement of production and movement of bile from the liver down towards the duodenum as well. And so that's going to feed back into there to allow that to occur. The gallbladder's job is to take that bile and store it. As it does so, it's going to absorb water. So it's going to concentrate the bile by getting rid of the water content there. And as long as everything is working properly, as long as we don't have excess amounts of things like cholesterol, we're going to have no problems with this process. If, however, the gallbladder essentially pulls out too much of the water portion, we get precipitate, we have the potential to develop gallstones. So we don't want to have that whatsoever. We'll come back to that in just a moment. So the bile is released in response to lipids coming into the duodenum. So as the stomach releases lipids into the duodenum itself, that stimulates the production and release of CCK and secretin from the duodenum. And that then again feeds back up into the gallbladder, feeds back up into the liver, and we get the secretion of bile salts, bile, into the small intestines. And that's going to help with all of our facilitation of absorption of lipids from that particular meal. This is also part of the process, or part of the reason why when we talked about the stomach and regulating kind of emptying of the stomach, that it wasn't just basically going from the stomach instantly into the duodenum, especially when there was fats there. It's going to take some time for that digestion to occur on there. And so the gallbladder has to release that bile and allow for it to move out of the duodenum in order for us to release more. So that cholecystokinin, or CCK, is going to cause weak contractions of the gallbladder. It's going to release the hepatopancreatic sphincter. So that's going to start to relax, and that's going to allow that to open up and allow all of that bile to enter into the duodenum once we've been stimulated to do so. That then allows us to have the food in the small intestines start to become broken down. And so we're going to start to do chemical digestion here in the small intestines. The small intestines itself is divided up into three portions. We have the duodenum, the jejunum, and the ileum. So three different regions to the small intestines that are there. And essentially it runs from one sphincter to another sphincter. And so we have the pyloric sphincter at the stomach, and then we have the ileocecal valve at the colon. And that is going to be the beginning and end of the small intestines themselves that are there. At the duodenum, the bile duct and the main pancreatic duct are going to have their opening, the hepatopancreatic ampulla. And once again, that control is the emptying of those two is controlled by a sphincter called the sphincter of Oddi. And so that's going to be relaxed when we have that presence of CCK. So that's going to allow us to release that at that point on there. So we start with the duodenum. The duodenum is the shortest portion to it, and then we have the jejunum. The jejunum, for all intents and purposes, is kind of in the upper left quadrant of the abdomen. And then we have the ileum, which is basically down in the lower right quadrant, making its way towards the... So here we have the intestines for the continuation of the small intestines. So we have the duodenum here in the first portion, and then we have over on the right hand side, we have the drawing, we have the jejunum, and then the ileum making its way down towards the connection of the colon that's there. From the outside, the small intestines don't really have any kind of special features that we can see. that's there. Basically just looks like a big long tube. But on the inside, what we saw in that last picture there was that we had some folding on the inside, similar to the rugae of the stomach. Here we have what we call plica circularis. And so those plica circularis are the individual folds that we have inside there. On those plica circularis, we then have villi. And so all of these little fingers here are the villi. And then on the surface of the individual columnar cells, we then have microvilli. And so all of this is all about increasing the surface area. So increase the surface area so that we can do absorption. Make sure that we're getting everything absorbed possible that is in there. So although to the naked eye it's going to look relatively smooth, in fact, when we start to look at it on the microscopic level, we have all kinds of infolding of the surface of the small intestines, all making it possible for us to potentially have absorption of every nutrient that we can. Different picture, but same kind of concept there. This one also helps to show the villi themselves and how running up the center of it, we've got our blood vessels as well. So we have a capillary bed running up into them, making their way up into each individual villus. And then remember that lipids require lacteals, the lymphatics, in order for them to actually be absorbed. So the majority of all of our lipids are going to run into the lacteals rather than going directly into the capillary bed. In addition to absorption, we're also going to have some secretion along the way of the small intestines as well. The cells that are there, for the most part, are absorptive cells. These are doing the absorption after things have been digested. Intermixed with them, there's going to be some goblet cells. And then deep inside those, towards the kind of the bottom of the villi, we've then got some enteroendocrine cells and some Paneth cells. So it's two different kinds of cells inside there, down deep. And the enteroendocrine cells are going to be our hormone-producing cells. So these are endocrine cells that are there. And then from there, we also have Paneth cells. And so once again, we've got resident macrophages. So we've got phagocytic cells that are running along the length of the small intestines as well. And so here we can see them down deep towards the bottom, kind of in between the individual villi themselves. And we have our intestinal gland. And inside there we have our enteroendocrine cells, our Paneth cells. Up further, we have our absorptive cells, so all of our columnar cells. And again, interspersed, we've got some mucus-producing cells. So we have the ability to produce mucus along the way as well. Keep everything nice and lubricated. Allow for the food to pass by without causing really any kind of damage to the inside of the intestines. As we move deeper into the small intestines, again, the only way really to kind of tell an individual segment apart from the rest in terms of the small intestines is to look at it under a microscope. And so when it comes to the duodenum, the duodenum has some special glands called Brunner's glands or duodenal glands. These glands are found only in the duodenum, and their job is to neutralize all of that acid chyme coming from the stomach. So they're going to produce an alkaline mucus that is going to help to protect the inside of the small intestines at the duodenum. Further on down in the ileum, we have what are called Peyer's patches. These are specialized regions in the ileum that are similar to lymph nodes elsewhere in the body. And so these are histologically different from the rest of the tissue that's found in that particular area, and that allows us to see some differences within the spaces there. A large amount of fluid is produced in the digestive tract. Just from the small intestines alone, we're getting anywhere between 1 to 2 liters a day. So a huge amount of fluid, which stands to reason when somebody has something like diarrhea, they are going to lose a large amount of water from their body, and if done so in a relatively short amount of time and they can't replenish those fluids, can very easily lead to dehydration. So lots of fluid present in the small intestines. In the small intestines themselves, this is where the majority of all of our digestion occurs. So carbohydrates and proteins, those began their digestion process up in the mouth and in the stomach, but only a limited number of types of carbohydrates have been digested, primarily starches. Proteins, small amount of digestion there as well. We've got some other protein enzymes that are going to help to break down them in the small intestines also. Up to this point, basically no fat digestion. A little bit in the stomach, but that's it. So we haven't really done any kind of fat digestion up to this point on there. The fluid that's entering into the small intestines is hypertonic. So we've got lots of things in there that is there. And initially it's coming in with a relatively low pH. So we're going to have to mix things around quite a bit on there. And all of that mixing, all of that churning helps to chemically digest all of the substances that are coming in from the meal. If we should happen to be missing or deficient in a particular enzyme, we're not going to be able to digest that particular substance, whatever it happens to be. That's there. So this is where things like the ability to consume milk products, somebody who is lactose intolerant, does not produce the enzyme lactase, and so it does not get digested. When we're moving through the small intestines, because there's so much mixing that needs to happen, the most common movement that we see is segmentation. So we're moving and mixing things back and forth and back and forth and back and forth. This moves our chyme, gets our enzymes mixed in with the chyme, and then most importantly, allows for the nutrients to be absorbed. They've got to touch the walls of the digestive tract. So if this is our digestive tract and we've got a piece of food here in the center, and that's some sort of digested component that's there, if it moves along the digestive tract, let me draw another section here. If it moves along the digestive tract, completely never touching the walls, it's never going to get absorbed. So that segmentation is going to squeeze in the sides, move that food around, and allow for us to hopefully absorb those particular nutrients that are there. After all the nutrients that can be absorbed in that particular section have been done, then peristaltic waves are going to move it on to the next section. So we're going to move it along, move it along, move it along. And now all of the leftovers, in a sense, from that meal get moved into the large intestines. So this is going to get continued to move along into the large intestines from this point, including any kinds of debris from our own digestive tract, things like bacteria that may have been starting to grow. All of that continues to move along. And it eventually enters into the large intestines. The large intestines is the last little loop that we have after absorption of essentially all of our nutrients has occurred. It does have some special features that we haven't seen anywhere else. On the outside, this is the first time where we're going to see something that kind of sets it apart from other areas. We have what are called Taenia coli. These Taenia coli are smooth muscle bands of longitudinal layer muscles that are running along the large intestines. It makes it look almost like there's a ribbon on the surface of the large intestines. They're very distinct. We'll see it here in just a bit. On both the inside and the outside, we see little pockets. And so we see that the large intestines has like these little bumps on it, and these we call haustra. These are the little indentations that occur along the large intestines. We also have, extending essentially off of where the Taenia coli are, we have epiploic appendages. And so these little epiploic appendages are adipose. And so we have little fat-filled pouches that are filled with adipose running along the large intestines as well. The food from the small intestines enters into the cecum. Extending down off of the cecum, we have the appendix or the vermiform appendix. And then we run through the three different portions of the colon, the ascending, the transverse, and the descending colon, and then the sigmoid colon, and that then gets us into the rectum, and then finally the anal canal for defecation. The cecum is essentially a pouch that is kind of below where the ileum actually dumps into the large intestines. So as food waste products are coming in, they come in and essentially dump into this little pouch that's there. Instead of it being a continuous loop or a continuous sac in a sense, there is this little out pocket that comes off of it. Extending off of that, we then have the appendix. And so that's going to have that extending off of it as well. So here we can see some of those special structures. So the ribbon here, that's our Taenia coli. There's three little portions of it in different areas along the large intestines. This little pouch here is a haustrum. And so we have individual little segments there. So you can see one there, there, and so on for those. And then these kind of like little feathery or leafy looking structures here, these are those epiploic appendages. So they kind of appear to extend off of the Taenia coli and kind of hang off of the large intestines itself. And again, you can see where the ileum dumps into the cecum there, and it is not just a continuous tube. So we don't have a full continuation there. The colon itself begins at the cecum there, and then we have the ascending colon as it turns. So if we have the ascending colon, we then have a curve, and then we have the transverse colon. The first curve is the hepatic flexure, or the right colic flexure. The next curve is our splenic flexure. And then we have our descending colon, so ascending, descending, transverse. And then we're going to have the sigmoid colon. And so the sigmoid colon is going to make a couple of curves as it makes its way into the pelvis, as it goes on down through there. And then we come into the rectum, and then the anal canal on the way out on there. Just like other places, we have to control movement. And so we have the ileocecal valve controlling movement into the large intestines. And then we also have valves that are going to control exit from the bottom of the digestive tract itself. So at the rectum and the anal canal here, we have a couple of different sets of sphincters that are there. In the rectum, we have so here's the rectum, almost what amounts to shelves in a sense, kind of these little outpocketings that are there. And these serve as valves that are going to essentially allow gas to move along and keep the feces back. So this what allows you to have flatulence that escapes without the actual fecal matter escaping as well on there. And then we have at the anus, we have two sets of sphincters. And so we're going to have an internal sphincter, and then we have an external sphincter. And so the internal sphincter is essentially superior. The external sphincter is inferior. The internal sphincter is smooth muscle, so that means it has to be involuntary. The external sphincter is skeletal muscle, so this one is voluntary. So before we are potty trained, the involuntary and voluntary sphincters themselves are under control of a reflex, and that is going to allow for them to relax when we need to defecate, and they're going to both relax at the same time. But once we become potty trained, the external sphincter becomes under conscious control, and we can essentially stop the fecal matter from coming out until we get to a toilet that's there. And so they relax only for defecation itself. Microscopically, the large intestines is primarily made up of simple columnar epithelium, except once we get to the anal canal. At the anal canal, the feces itself is going to be relatively dehydrated. So we're going to pull out a fair amount of liquid along the large intestines. That's his primary job, is to reclaim all that water. Get back that 2, 3, 4 liters of fluid that we've dumped into the system along the way. So by the time we get to the anal canal, things are going to be dried out. So we're going to need some protection. Part of that's going to come from goblet cells. Part of that's going to come from stratified squamous epithelium. That's there. And so within that, we have a little bit of protection there. The anus itself has mucus-producing cells there as well, and it's also going to help within the rectum and the anus to essentially compact the fecal matter itself into one component that can be released. And so we have them leave at that point. Lining the sides of the anal canal itself as well, we have veins, and those veins are the hemorrhoidal veins. And so the hemorrhoidal veins that are there are unprotected. So back when we talked about the blood vessels and creating a situation where in the legs we ended up with varicosities, we can get the same thing happening here as well. And so again, kind of think about, we have a tube, and then on the running just deep to that tube, we have some blood vessels that are there, and there's nothing pushing against them. So there's no protection there in a sense for those vessels themselves. And so they can potentially develop into what we commonly call hemorrhoids that are there. And so the vessels themselves become stretched out, they become oblong, and that leads to the potential for hemorrhoids there. And here we can see those individual veins themselves. And again, there's no protection on the inside here of anything kind of pushing back, just like you have in your legs that are there as well. So there's no force that helps to help to drain those vessels. The digestive tract itself is a multitude of growth of different types of bacteria. And so we have bacterial flora going essentially all the way from the mouth to the anus. Even in the stomach, we have the potential for bacteria to grow there as well, in which we can have Helicobacter pylori. So there is even bacteria that can live in that low pH of the stomach as well. The amount of bacteria that is there is going to be greatest in the colon itself. So we do have a larger amount of bacteria that lives in the colon that is there. Some of that bacteria we actually need. And so some of that bacteria is there for helping out with our ability to elicit different micronutrients from our diet. And so B vitamins, an important one, vitamin K for coagulation, things like that are being produced here. The downside is that as bacteria do their thing and they consume different types of substances in the colon itself, they produce gases. So whether it's bacteria, whether it's different forms of yeast in the intestines themselves, they're consuming any leftover carbohydrates. And that's again where things like our sugars in milk, we have the potential that if that lactose is undigested completely by the time that it gets to the large intestines, there's a huge amount of now food for bacteria to start to consume and then multiply, and that causes all of the symptoms that go along with someone who is lactose intolerant. So as I said, the primary goal of the large intestines is water reclamation. So we're trying to get back as much water as possible that is there. Those bacteria are producing some nutrients for us. So we want to get some of those vitamins that they're liberating for us as well, and any other leftover electrolytes that may have been in the food up to this point as well. From there, move it down towards the rectum, let it sit in the rectum for a little while and become compacted before it's ready to leave the rest of the body. The large intestines has those big, long tinea coli, and then it has circular muscles in each of the haustra. And so we get segmentation of the large intestines as well. This essentially contraction moving along the large intestines themselves helps to move the food into the next region. So it's essentially pushing it along, contracting. As it stretches out, it pushes it into the next segment. That next segment then pushes along the next segment, and so on. And so we continually push that food along there. One other type of reflexive contraction that we get is as we consume a new meal, we also have what we call the gastrocolic reflex. And so this is going to help to essentially clear the last meal through the intestines and down towards the rectum, essentially making way for the next meal that's coming in. Once that waste has been into the rectum long enough, it's eventually going to essentially pile up, so to speak. And as it does so, it starts to stretch out the rectal walls. And as those rectal walls stretch out, that sends signals up to the spinal cord. And then the spinal cord sends back down signals to the sphincter muscles. And again, if we are an infant, both the internal and the external sphincter muscles are going to relax and defecation is going to occur on there. If we're potty trained, then we have to consciously relax that external sphincter. So here we have distension occurring into the rectum itself. That sends up signals up to the spinal cord. That sends signals down to the internal sphincter. The internal sphincter relaxes, but we also need to send down conscious control relaxation to the external sphincter muscles to allow them to relax as well. And when that all happens, then we get defecation. Back when we were talking about the small intestines and the production of bile, we talked a little bit about lipids and how lipids are going to be absorbed primarily through the lymphatics. And so they're going to make their way into the lacteals that are there. And with that is the majority of all of our lipids are going to have to be packaged up. So when you think about it, blood plasma is water. Water and lipids don't mix. And so we have to surround all of our lipids with essentially a coating that allows us to actually move those lipids into the bloodstream eventually after they go into the lymphatics that are there. So chyme moves along that are there, and the bile salts create these micelles They basically surround the lipids, the myoceles themselves, move up to the lining of the small intestines, and release their lipids into the lymphatics. From there, those cells are going to form what we call chylomicrons, and those chylomicrons, as they went in through the absorptive cells, dump the chylomicrons into the lacteals, and we now have absorption. Lots of things go into the digestive tract. Some of them may be harmful over time, and so different things have the potential to start to form cancerous cells, whether it be in the esophagus, whether it be in the stomach, the pancreas, the liver, small intestines, large intestines. They all have the potential to develop cancer. In the United States, colon cancer is the most common of the digestive tract cancers that we have. That's there. So it is high up there on the list of all cancers that are found. And the downside, in a sense, besides that it's cancer, to digestive tract cancers is that oftentimes they are caught later on. Because many of the symptoms, constipation, diarrhea, pain, blood in the stool, may be missed. We don't really talk about the digestive tract. We don't talk about bowel movements in terms of in polite conversation. Those kinds of things aren't oftentimes looked at. And for some people, thinking that they're constipated or have diarrhea often is just what happens. That's just what they do, in a sense. But it is not normal on there. And so things like regular colon testing after the age of 50, colonoscopies, even some insurance companies do things like home screening before the age of 50 on there. If there's family history of colon cancer, then obviously screening in terms of a colonoscopy is warranted earlier on as well. One of the things that things like home testing or simple testing in the doctor's office looks for is what we call occult blood. And so we're looking for detectable red blood cells in the fecal matter that is there. And this is essentially an indicator that potentially something is going on in the digestive tract. Somewhere there's blood that's being lost into the digestive tract on a daily basis. If it's happening high up, say a bleeding ulcer, it's going to become digested along the way and can actually lead to things like black tarry stools. And so they look like essentially black tar in terms of the fecal matter that's coming out because it is digested blood along the way. That's there. And with that, that concludes our look at the digestive tract, and hope you come back again later.